So good afternoon, everyone. I wanted to tell you a little bit about the project that we are currently running here at the Silesian University of Technology. The project is called Flow-char. It's funded by the Norway grants via our Polish National Center for Research and Development. And in this project, we want to combine biomass gasification, which you're all familiar with the water desalination using the technique called flow-electrode capacity deionization. So the main idea is to use the biochar from gasification as this flow electrode. So I will start with saying you a few words about the FCDI process, because I guess it might be new for at least some of you. So starting with a classical capacity deionization that uses porous solid electrodes. This is a process that you can use to remove ions from water, for example, sodium and chlorine from salt water during the desalination. And here you can see it's basically an electrochemical process where you applied electrical potential to the current collectors. It could be graphite or metal plates that are attached to your porous electrode. It's quite often activated carbon that's used as the electrode material. And so when you apply the potential and you charge these two electrodes, when you insert a water flow between those electrodes, then the cations and anions will be electrosorbed on the surface of the charged porous electrodes. And so at the outlet of the reactor, you can get a clean water. With this process, with the solid electrodes, after some time, your electrodes will get saturated with the ions. So inevitably, after the adsorption or the charging step, you need to disconnect or reverse the potential and do the desorption step, where the ions get back into the water stream. So during this discharge step, you are collecting the concentrated brine as a waste from the desalination process. Recently, there's been a twist to this method. So this so-called flow-electrode capacity deionization. The trick here is that instead of a solid electrode, you have a carbon suspension, carbon particles suspended in an electrolyte. So this is the so-called flow electrode, and it's flowing through the dedicated channel. So you have two separate channels for each electrode flow. And these flowing electrodes are separated from your main compartment where the water is flowing, with the ion separating membranes. So here, when you charge your electrodes, the ions are just traveling through the membrane into the respective cathode and anode. This way, you can run the process continuously because you can discharge your electrodes outside of the reactor. So this is one of the benefits of using this type of module. Although for the sake of the project, we are now running it in an isolated cycle because we want to study the spent electrodes, see what happens to the carbon after the process. That's why we want to collect them without discharging them. So in our case, it looks like this. You have one electrode and the other electrode circulating separately. And also the water that we desalinate, we are recirculating right now because the module is small, so running it in single pass wouldn't be possible at the current state. But if you assume that you can stack the modules to get a bigger contact area between the electrodes and the water flow, you could run the process as a single pass also. So to show you how it looks in real life in our lab, this is our module. you can see it's not that big. The contact area is around 20 square centimeters. All the rest of the stuff around is just the equipment to run it. So the peristaltic pumps, DC supply, current supply, and everything. And if you look inside the module, the most interesting parts are these graphite current collectors. So the plates that we apply the potential to, and you can see that they have this carved pathway inside them. So this is this small serpentine. This is the path through which the flow electrode, so the carbon suspension, flows. It flows through an inlet and then goes all the way up onto towards the outlet of the module. So here is where the electrode flows. On top of it goes the membrane, one of which you can actually see here bathing in the electrolyte solution, waiting to be inserted into the reactor. And then, interestingly, this really thin plastic separator that you can see here, and this net that is fitted inside, this is the water chamber. So where the water is flowing between the flow electrodes. So if our pathway is around 2 millimeters deep, the pathway for the flow electrodes, the chamber for the water flow is 0.5 millimeter wide. So it's really thin. I guess it's nothing surprising for any electrochemist working with these processes, but for me, coming from the biochar background, it was kind of interesting that it's such a thin layer of water. Basically, you want to reduce the resistance and also make the shortest possible pathway for the ions to come through the membrane into the electrode. So you need to keep this chamber really thin. Okay, so this was like an introduction about the FCDI to just give you up to date what we are doing in the lab. And then going back to carbons. As I've mentioned, quite often the flow electrode is made out of the activated carbon particles, the commercial carbon. So what we want to do in this project, we want to see whether some sort of a pyrogenic biochar can be used instead. And as you all know, working with the biochars, they are sometimes quite tough to characterize and to work with, especially if you want some more sophisticated application for them. So you don't really activate them in any particular way. The surface area is rather smaller than with the activated carbons. They are more microporous. They are highly heterogeneous with no special functionalization and with high inorganic content. So the only way that they can, the only advantage that they can have compared with the commercial products is just their low cost and well, environmental friendly way of getting them as the byproducts and not especially prepared stuff. So with all the biochars available, we wanted to focus on the gasification ones, just to have the highest surface area that we could get out of these materials. Because as you all know, the gasification is working a little bit like a thermal activation of the carbon. You can get more porous particles, and because the porosity of this of the carbon used for flow electrodes is quite an important, or one of the most important aspects of the working electrode. At least it's considered one of the most important aspects. So this was the feature that we wanted to focus on in our preliminary studies about the material that we want to use. So just to briefly remind you about the pore sizes that are distinguished based on IUPAC report. With the micro and mesopores, this is the area that we are interested in. We measure it with gas adsorption methods. We are especially interested in super micropores and in the smaller mesopores because these were supposed to be the most important ones for the ion absorption. And to determine them, we've been using DFT models, which basically relies on fitting your gas absorption isotherm that you experimentally measured with the theoretical isotherms and with the numerical methods and regularization procedure. You can obtain the representation of the pore distribution in the in your samples. So we've been doing this with the dual to the NLDFT model aimed at heterogeneous carbons that was developed by Jagiela et al. And in this one, we use nitrogen isotherm and we couple it with the CO2 absorption isotherm because this way you can skip the low pressure part of the nitrogen isotherm, which is usually not very accurate and also really hard to measure. So you can use just the higher high pressure part of the nitrogen isotherm, couple it with the CO2 absorption isotherm, and then doing the simultaneous feed to both kernels. You can get your calculated isotherms based on which you can get the pore size distribution. We think this method is particularly good working with the microporous samples because the commonly used PETs often underestimating the surface area of the sample. If it's a more wide distribution, the difference is still significant. But if you use a very ultra microporous materials, you can see that because you don't use CO2, you just use nitrogen absorption, the PET value is really low, and you can quite a lot of surface area still hidden in the ultra micropores of the samples. So we chose this way to evaluate our samples. We used it previously, for example, for sewage sludge, where we studied the pore development in during gasification with CO2 and steam. So for this research, we didn't use sewage sludge. We used chars from agricultural and forest residues. We tried to select a few examples of different biomass groups. Here you can see the pore distribution in the pyrolyzed chars before the gasification. We do the char preparation in 2 steps. So we first pyrolyze them, and then we try the gasification with different parameters. So you can see that the olive stones and beechwood here, which actually even after pyrolysis they have quite a wide pore distribution. For the main campaign with selecting the final gasification parameters, we've used walnut shell biochar because the distribution was somewhere in between the most developed and the really bad ones. So we figured we could use it as sort of an average of our biomass chars. And we tested with different gasifying agents. So using nitrogen as a balanced gas, we used lower and higher concentration of steam. We tried steam with CO2, just the CO2 and small amounts of oxygen. Most of the tests were carried out at 800 Celsius degrees. We did 3 tests with elevated temperature as well, just to see whether it's gonna affect our pore distribution. And so here you can see the distribution of different pore types in the char correlated with the burn up of the char. So we ran the gasification for different times, residence times, getting different char burn off. All the tests that were carried at 800 degrees with steam are indicated in blue, and you can see some trends emerging from this tests. Interestingly, at 1000 degrees, even the measurements that were using steam, for example, these two that you can see here, they didn't really follow the trend. Also, using only CO2 or using oxygen didn't manage to develop super micropores and mesopores. The ultra microporosity was behaving pretty much the same for all the samples, so it was just slightly decreasing with time. But based on this, we decided that we definitely need to use CO2 as high concentration as possible, and also limit the temperature to this 800 degrees, not going any further. So this was the gasification parameters that we used for char preparation. And besides walnut, we also prepared the char from olive stones and beechwood, because as I mentioned, they had the promising for distribution even in the pyrolytic chars. After the gasification, you can see that in both the samples, the porosity was developing quite nicely, even including some mesopores. So they looked pretty good. And when we compare them with the walnut shells, the trends are similar. So the decrease in ultra microporosity, increase in other pores. Interestingly, there was a different ratio in super micropores and mesopores in walnut. That was walnut shells that were yielding rather super micropores in the char, and the other samples actually went all the way to mesopores. Besides the chars that we prepared in the lab, we also tried some materials that we could use as a reference to compare the laboratory scale chars. We also used the material from the semi-pilot scale gasifier that is operating at Technical University of Denmark. This one was a char prepared from the wood, spruce wood. You can actually get more details about the char itself in works of Julia Raveni. She was studying this char for quite a long time and has some quite extensive characterization of it. And as the reference for the commercial carbons, we used Norit Supra from Sigma Aldrich that has been commonly used for FCDI purposes as well. So we just wanted a reference to see how the biochar works compared with the commercial products. And here you can see some tests from the saline water desalination. So you can see the decrease in the salt concentration with time as the process was going on. Here are the chars that we prepared in the lab. The walnut shells was working slightly worse, and for these three chars, the dependency that the wider pore distribution, more mesopores, is a beneficial thing. We actually could see this trend for the chars that we prepared. Because the olive stone and beechwood char were working better. But then if you look at Norit, which has really a nicely developed porosity in all pore sizes, it didn't really help it with water desalination tests. And most interestingly, the so-called Viking char that didn't really look like that, didn't really look that impressive in terms of the pore development, but basically worked in half the time of the commercial carbon during the desalination tests. So now we really need to focus on finding what's the reason behind the different efficiency of the chars that we are using, because it's not solely to the surface development. We believe it's also gonna be dependent on the conductivity of the chars and also on the surface chemistry, so functional groups, inorganic content, For now, we've just this is also the table comparing the volume of the pores. You can see that Norit has quite a lot of super micropores and mesopores compared with the other chars. And for now, going back to the surface area, we've managed to look at the FTIR spectra of these samples, and the chart from the pilot scale gasifier differed from the other ones significantly. It had this peak here at 1400. It might be some sort of a resonance stabilized carbon-carbon or carbon-oxygen bonds. Either organic or inorganic compounds on the surface of the char. This larger peak here, it's also either heterogeneous oxygen heteroatom oxygen heteroatoms in the structure of the char, or some inorganics such as silica. So we definitely need to dig deeper into the chemistry of this materials. So for the next steps, as I mentioned, electrochemistry to see the resistance, the capacitance of the char, and then hopefully some digging into the morphology and chemistry of the chars to see what's behind their performance. We also, as the next step of the project, we are currently studying the kinetics of the spent chars, because one of the aspects of the project is also to return the spent electrode back to the gasification process, just to get rid of it after it's not possible to regenerate it any further. So yeah, basically this is the work that we've been doing so far. And with this, I'd like to thank you for listening, and also once again acknowledge the founding body. Thanks.
Thank you very much for this very interesting presentation, especially for someone who has never heard of flow electrodes before. And also thank you for explaining the process distribution, because I'm very interested in that, and you almost always only see the PET surface area, which is a bit useless in many cases. So thanks for that. So if you have any questions, please write them into the chat. My first question would be, maybe I missed it, but how did you paralyze and gasify your samples? What was the reactor?
Yeah, so the pyrolysis was actually done in a muffled furnace in a closed crucibles, the ones that you use for volatile matter determination. Because with the short residence times, we didn't really see any oxidation of the sample, and we were about to gasify it anyway. So we didn't really go for the closed quartz furnace. But for the gasification, it's a vertical quartz furnace with the externally electrically heated furnace with the quartz reactor, char bed suspended on the quartz wood plug, and gases inserted with a certain composition.
Okay, thank you. And the Kochevisa maybe was asking, hi, thanks for your great presentation. I have a question. Could you share a photo of your gasifier? Also, what coating and material in the gasifier is made of? So what the I'm not sure. Do you mean the commercial one or Kochevisa?
Or the laboratories?
Do you have a picture of the? I guess it's only the publication from Julia.
I could have a look whether I can find something. Just wait a second.
Because the other gasifier, as we just heard, is a quartz glass vertical.
Yeah. So you mean the laboratory scale gas? So the reactor that we do the gasification in the lab, or the?
I think we have to wait until we get a question more specific. But you got one more question. So maybe we discussed it first. So in the desalination tests, did you add the char in the same mass concentration for all chars, or did you add them in a way to get the same specific surface area equivalence in the suspension?
In the suspension, it's mass concentration. So we use 5% char in electrolyte.
So did you have a lot of different densities from the chars?
We didn't get to the densities yet, but it will differ, depends on the porosity. That's for sure. But usually with all the electrodes, you aim for the same mass, and obviously then if you have a more porous structure, then it might be more of it volumetrically. Although the particles are cryomilled to the very fine powder around say 10 micrometers or something like this. So the biochar is quite dense anyway, at least in terms of the particle size.
And you, so Coach Visa was specifying the lab scale reactor. You wanted to see if you have a picture. If not.
Yeah, I can show you some of the old presentations here. Wait. Okay. So this is more or less one of the reactors that we've been using. Here, actually, this is shown in the mode where you can continuously weight your sample during the reaction. But in a more typical arrangement, it's a closed reactor, so the char bed is suspended. But this is the reactor. Here there is a water evaporator where we insert through the syringe pump. We insert water to evaporate it to create water vapor, and all the gases are just flowing through this evaporator. So all the mixture, pre-prepared mixture, is then traveling through the main reactor. This is yeah. So this is another type of reactor. We actually wasn't using this one. This was like the first smaller one used for previous tests in the past, but it's again similar principle. So basically, the one furnace where you evaporate water that you inject and supply all the other gases, and then all of it is traveling through the bed of char that's been. This was in here. It was used for volatile conversion, but basically, if you forget about the toluene, then this is more or less how it looks like. So you're basically preparing your mixture, gasifying your char bed, and take it out after the reaction.
Okay, great. Looking at the time, I think we have to stop here. You got another question from Vera in the chat. I think your colleague, probably Hannah, was already starting to answer some of them. But if you have some minutes, if you can have a look at the chat, that would be great. So yeah, thank you again for your great presentation. Thanks a lot.
Thank you.
And we